Okay, welcome back everyone. At this time, I would like to introduce Dr. Ravi Kulkarni. He is an assistant professor in the Department of Population Health and Pathobiology at North Carolina State University. The title of Dr. Kulkarni's research project is Enhancing Immunogenicity and Protective Efficacy of Recombinant Infectious Laryngotracheitis Vaccines in Broiler Chickens.
Thank you. Good morning, everyone. Thank you for joining. And today I'm going to talk about one of the USPOULTRY Foundation funded projects, which is about the infectious laryngotracheitis virus. And then we worked on specifically how we can enhance the immunogenicity and protective efficacy of these recombinant vaccines. So what I'll do is next about 15 to 20 minutes or so, I'll start off with a quick intro to the disease and also identify what was the problem, why did we do this particular study. And I'm going to show you two studies. They are not mutually exclusive, but they are kind of connected, and then I'll walk you through the results as well as the main findings of this work that we have found so far. So to get started, infectious laryngotracheitis is a DNA virus, and we all know that viruses are of either RNA or the DNA. Classical example of the RNA is the coronavirus. We all are well educated. On the DNA side of it, the ILT, which I'm going to refer to infectious laryngotracheitis in the rest of the part, it's a DNA virus, and it's a highly contagious respiratory disease and very economically important. And the other thing is that this is a DNA virus and also susceptible to commonly used disinfectants in the farm environment and so on. So the way it spreads is horizontal transfer, which is bird to bird through the aerosol routes, through the droplets. And the one that you see in the picture it's basically shows the how the structure of this DNA virus looks like, with a lot of the surface proteins. One of them is also glycoprotein and so on. So clinically, as I said, if it is affecting the respiratory tract, you will have the respiratory distress that we often see in the birds, and particularly affecting the trachea of the birds in this case, which causes the coughing. And the virus, as it replicates, it affects the mucous membrane, and then the blood also comes out. So that's why sometimes coughing is also mixed with blood or a bloody expectorant. And we also see that the eyes become watery, and then it is a typical case of conjunctivitis, which is seen in ILT affected birds. And mortality is very variable. It could be low or as high as 50%. And particularly in the layers, it reduces the egg production. So some of these pictures that are shown here, as you see that there is blood expectorant here, there is conjunctivitis, and you see this poor bird gasping for air because the gross lesions that are seen when you cut open the bird, look into the trachea, is particularly you see this very reddened mucosa. There is blood in the trachea, and over a period of time, the mucus mixed with the blood in turn, resulting into an exudate and dries out, causing the mucosal plug. So right at the trachea, the caseous plugs are formed, due to which there is respiratory distress, and then we saw the bird gasping for air. And sometimes the lungs are also get affected, and you see is reddening of the the lung tissue. And sometimes we also see the exudate right at the point of bronchi. So now we know generally that it is well controlled using the vaccines, and there are two major types of vaccines. For simplicity, modified live vaccine viruses and the live recombinant vaccine viruses. So of the live vaccines, we have one called the chicken embryo origin, CEO, which I'm going to refer to as, and the other is tissue culture adapted, which is TCO. Whereas the recombinant vectored vaccines, the way they work is that there is a host virus, in this case either the HVT here as shown here or the fowlpox, which carries the other proteins, heterologous proteins, in this case the ILT or the laryngotracheitis protective antigen, so that the recombinant virus, when you are giving it, you can get two dual effects. One is immunizing against the Marek's disease, if it is HVT, or the fowlpox in that case. At the same time, you can also produce immunity against the ILT. So for this particular work that I'm going to show here is we have focused on the CEO as well as the recombinant HVT-LT. Now, every vaccine has its own advantage and disadvantage. The CEO, the best part of the CEO is it's a very effective vaccine. It can protect against the clinical disease, prevent mortality pretty significantly, and it's a more robust protection when compared to the other vaccines. However, every advantage comes with sort of a disadvantage. In this case, CEO, since it's a live virus vaccine and it is replicating and then being shed to other birds, so during this bird-to-bird transmission, the non-pathogenic or the attenuated virus regains the virulence and then becomes a problem in the farm. So that's why we call there is a spillover effect to other farms, and there is prolonged shedding of the virus, which also results into seasonal outbreaks. Now, what about the RHVT-LT, the recombinant? The best part of it is that they are safe, can be administered in ovo for producing an uniform, the immunity, and it also prevents the clinical disease to some extent, improves the production parameters. And unlike the CEO, this doesn't carry any spillover effects or regaining the virulence and so on. However, the problem is that they are poorly immunogenic. Immunogenicity is the ability of an antigen to produce immune response. So the immune response is inadequate, which leads to an inadequate protection, and it's not as good as CEO. So with these, now for any research, you need a strong hypothesis. In this case, what we hypothesized that the recombinant ILT vaccines that have some deficit in terms of the immunogenicity or the protection ability, if we adjuvant this vaccine with a known molecular adjuvant, we could boost the immunogenicity as well as the protection ability. So what we did was we used a molecular toll-like receptor ligand adjuvant called the CpG DNA. If you're wondering why CpG, it's based on my previous experience while I was in the vet school Guelph in Canada, as well as postdocing at Emory Vaccine Center here in Atlanta, which worked very well with my experience. And then we added the CpG DNA and hypothesized that it can help very well. So now the CpG DNA, it is a ligand. It binds to a receptor called the TLR21, which is located inside the cells of the immune system. As you see here, there is TLR21. The CpG DNA, when it is taken up by the immune cells, it triggers the immunostimulation, leading to cytokine expression and a good immune response. So when you are incorporating an immunostimulant with the vaccine, you should be able to see an enhanced immune response to the vaccine too. So the design is, I have tried to simplify as much as possible, is when we have the CpG DNA, we want to put it with our HVT-LT and then put it into the eggs. In ovo, one thing is that dose optimization is important. We chose 5, 10, and 25 micrograms of CpG DNA and asked the question whether it affects the vaccine virus replication. As you see here on the right-hand side, 25 was a big no, and 5 and 10 were pretty good. And then the other thing is now, will it affect the hatchability? In terms of the hatchability, as you see here, I think these numbers are pretty microscopic. You have to believe what I say. Here, when we look at the 5 and the 10, the hatchability was not drastically affected. So we thought that 5 and 10 are doing good, and then we immunized or in novo vaccinated with this particular formulation. And the first thing what we did was we took the day-old hatched birds in order to see whether 5 is better or 10 is better. In order to do that, we took those day-old chicks and then removed the spleen, and then we did some immunological work, which included the gene expression studies here, and we also looked into the cellular immune responses, which I will explain to you in a bit. So before that, I thought, looking at the audience, I'll give a quick intro to when we talk about various immune responses in the next set of slides or so, is basically an immune response is divided into innate and adaptive. Innate is something which comes on very immediately. It comprises of cells such as macrophages, dendritic cells. They are ready. They are the first line of the combat. And then while they are fighting the virus, they also pass on the signal to the other component, which is adaptive component, which is a more systematic, well-organized, it's a good immune machinery. And then there are also some cells which are gamma delta T cells, which sit somewhere in between that, they can fight by themselves. At the same time, they ask the B cells to produce antibodies. And importantly, there are also something called T cells. And I would like to take your attention to these 3 or 4 important cell types, because rest of my presentation is going to talk about these cells. So we have macrophages, gamma delta T cells, B cells, and then there are T cells. And again, I would like you to remember that there are 2 types of T cells, CD4 and CD8. You need T cells for an effective antiviral function, correct? So how do they do that? This looks really nice on the cartoon. How do they communicate? They communicate through producing some cellular, chemical messengers, which we call in immunology as cytokines. This is the cell. You see these tiny, tiny yellow dots. They are the cytokines. And then this is how they talk to each other, that, look, there is a virus. You got to get ready. Who needs to do what function and so on. However, there are a lot of cytokines, and all that I want you to remember is 2 important cytokines, interleukin 12 and interferon gamma, that are produced by T cells as well as the macrophages and other immune cells. So IL-12 and interferon gamma are the 2 cytokines which specifically mount an effective immune response against the viruses. So with that background, I'll show you what we found in our first study is that when you had a group of the 5 and the 10 microgram induced more interferon gamma and also the interferon gamma in the lung, which was a good news for us that we were able to choose that the 10 was doing good. And this is something where I want to introduce you to flow cytometry, is a technique where actually when you grind the tissue or put the blood into it, you can see the individual cells as they are seen here. There are millions of dots. Each dot represents 1 cell. So the way we work is that, we have these cells, run from the machine and then try to gate. You see this rectangular box, which is the live population, because you want to look into the live cell responses. And then we ask the question from the live cells, how many of these are macrophages, depending on the groups that we used. As you see very clearly that the 10 micrograms had very high level of macrophages that were activated. And then similarly, we also do a gating to find out which ones are gamma delta T cells, which shows that the 10 micrograms was again better than the 5. And if you remember that slide I showed you about T cells, remember there were CD4s and CD8 T cells. As an immunologist, we need to look at what were the different subsets of the CD4 and CD8 T cells, and obviously the 10 micrograms did a very good job on CD8 cells, And one more point here to mention to you that it's just, if there are more cells, it doesn't tell you there is a good immune response. You need to make sure, are they activated? Are they, ready to fight and produce cytokines and produce the immunity? And we saw that the 5 and 10 were both equally good. Altogether, the story led us to a strong conclusion from the first study is that the 10 microgram dose does a good job. So we published this paper in Vaccines, and then we moved on to the second study, which I'll finish in the next few minutes or so. So now we had 10 microgram dose, and we wanted to put it into Innovo and then ask the question that, in the previous study we had 1 day old, but here we want to see that does it really protect against infectious laryngotracheitis challenge. So for which we did this RHVT-LT along with the CPG and the chicks hatched, and then put them into the BSL-2 facility. And then we had many groups, which I'll talk to you briefly in a second. But before that, when we see that, this adjuvant is, whether in order to see if this adjuvant is working, you need to have something to compare with. So we had the CEO vaccine, which was given in the water at 14 days, which was known to produce a robust immunity. And we wanted to have, what our CPG is doing to the recombinant vaccine. So in the step 2, if you see that, there are 9 different groups. And what I see here is, I want you to focus on 3 important groups in order to look at the immune responses. The one which is green here is the one that did not receive any vaccines, but it was challenged with the wild type virus. So second is the group number 4, which is in red rectangle, is the CEO vaccine, and the group 9, which is in blue, shows where we had our CPG added adjuvanted vaccine and then also challenged with the virus. So what were the results? Here is the results. So what we did was that, the challenge was given with a wild type ILT virus at day 28, and then over a period of 5 to 6 days, they were clinically monitored and also did a necropsy to get with something called ILT index scoring. And this graph straight away shows you that there is one group which is CEO gives a robust protection in terms of ILT, which is really well known, and we were able to reproduce in our disease. But our more interest was in this group where we had the recombinant HVT-LT added with the CPG, and you can see that that also showed significant protection when compared to the which was unvaccinated group. So that was a good news. And then immunologically, we wanted to see how are these vaccines doing this, kind of a magic in terms of protection. We looked at the interferon gamma. Remember, interferon gamma and IL-12, 2 most important cytokines for antiviral defense. And we found that both the CEO as well as the adjuvanted group had higher interferon gamma, and the CEO, in addition to that, had higher IL-12. And then when we looked at the cells, remember gamma delta T cell, it's an innate T cell playing an important role in the antiviral defense. We found both the groups, CEO as well as the adjuvanted group, doing a great job in terms of enhancing the responses by the gamma delta T cells. And what is most important, if you remember, is the T cells. So if you pay attention again onto the group 4 and 9, and in this particular case, when CEO was given, we found a particular cell type, which is the CD8 T cell, got activated very well in order to fight against the virus, whereas our adjuvanted vaccine, we saw that it's a CD4 cell type. So that's why, as an immunologist, I think the CEO providing a robust immune response or immunities partially or more largely because of the CD8 T cells, whereas our CPG was able to induce good amount of decent, significant protection, which was because of the CD4 T cells. Now, with that, the conclusions were very crystal clear. The story began with starting with 5, 10, 25 micrograms, drilled down to 10 micrograms, went into the birds, gave it to them to look at the immune response, and it looks like the adjuvantation of the RHVT-LT with the CpG DNA as an adjuvant, we can really boost the immunogenicity and the protective efficacy of this vaccine, which was the main objective of this study. So for any research, for a researcher, there are two lifelines. One is you need a funding, and I really thank the US Poultry Foundation for this particular funding, as well as I'm also thankful to my college at NC State for providing the, the startup funding for the investigation. And important people is the second lifeline. Isabel Jimeno, who is who works with Marek's disease and also herpes virus expert, was a co-investigator, and Dr. Faisal Abdul Karim at University of Calgary was a collaborator, and the people who were really active on the bench, and significant contribution was made by Carissa Gagan right there. So she did quite a bit of work here, and then she has started a PhD with me in my laboratory. I also was lucky to get Matthew Browning, a brilliant, very hardworking DVM student in my lab, and also Abdul Hamid, who works with Dr. Jimeno's lab, was also contributed to this particular disease. So with that, I think I'm well within the time, and I can take any questions you may have.
Any questions for Dr. Kulkarni? All right, thank you very much.